Hi everyone. For exam 2, I have chosen the genetic mapping prompt. Here we have a potential genetic map for the phenotypes of fur color, fur length, and height of the poodle. The alleles show us that the golden poodle is dominant to the black poodle, that short fur length is dominant to long fur length, and that the standard height poodle is dominant to a miniature poodle. In this first test cross, we will be conducting a cross of parental generation homozygous dominant poodle and homozygous recessive poodle to create the F1 generation. Once crossed, this is what the potential chromosomes of the F1 generation would look like. Here, the F1 generation is heterozygous and obtains the phenotypes of the dominant parent. With the determination of the F1 generation, a heterozygous offspring, we go forward to cross it with a homozygous recessive offspring, which will become known as the F2 generation. This is done to determine the potential of there being a variety of genotypes and phenotypes within the offspring and its probabilities. Through this, we discovered two potential outcomes, known as crossovers. The first potential crossover is known as single crossover 1, which occurs in the R1 section of the genetic map. As seen here, the genotypes of the single crossover 1 are little c, big L, big H, and big C, little l, and little h. Also seen here are the genotypes of the single crossover 2, which are big C, big L, little h, and little c, little l, big H. The double crossover of the F2 generation comes next. This is where a crossover occurs in both the R1 and R2 regions of the genetic map. The genotypes for this offspring cross are little c, big L, little h, and big C, little l, big H. Now back to the genetic map. As you likely notice, there are distances between the sectors, R1 being 9 centimorgans and R2 being 13 centimorgans. These distances resemble the likelihood of a potential crossover to occur. So, as you may assume, the larger distance means that there is more of a crossover possibility in R2 than there is in R1. The coefficient of coincidence is 0.74. Here is the breakdown of how to find the probability of double crossover, abbreviated as DCO. Here is how to determine the probability percentage of single crossover 1. How to determine the probability percentage of single crossover 2 is shown here. You can find here the probability of no crossover occurring. This chart displays the phenotypes, genotypes, probability, and event type for each crossover. You can see that a double crossover event has the lowest probability, while no crossover has the highest probability percentage. Additionally, because the R2 distance is greater, that explains the higher probability percentage compared to that of R1. I hope that you have found this video to be informative and helpful. Thanks for watching.